Chapter 7, Photosynthesis. Why does photosynthesis matter? Nearly all life on Earth, including you, can trace its source of energy back to the sun. We humans can trace every morsel of our food back to plants. By capturing the energy of sunlight and using it to create organic materials, plants performing photosynthesis feed the world. Want to do something simple to combat global climate change? Plant a tree. You'll be glad you did. A solar revolution. Rooftop solar panels are becoming a common sight. The electricity which is produced by the solar panels using sunlight can either be used directly or can be used indirectly by transferring it to the national electric grid. Living organisms that can capture sunlight and use it to produce energy sources such as plants and any other organism that can do photosynthesis, these are all producing renewable sources of energy. Photosynthesis is a process whereby light energy is transformed into chemical energy by plants, algae, and certain bacteria. During the process, carbon dioxide and water are used to make sugar molecules, and oxygen is released as a byproduct. Autotrophs are those organisms that can generate their own organic matter from inorganic raw materials. Plants and other organisms that do this by means of photosynthesis, they are known as photoautotrophs, and they serve as producers for the ecosystems. The figure here shows some photoautotrophs. Chloroplasts are the sites of photosynthesis. They are found in the cells of leaves of the plants, and their green color is due to chlorophyll, which is the pigment that can help absorb the light, and this is the one that plays a very important role in converting solar energy to chemical energy. Chloroplasts are concentrated in the inner cells of the leaves, with a typical cell containing about 30 to 40 chloroplasts. Carbon dioxide enters a leaf and oxygen exits by way of tiny pores called stomata. Singular would be stoma. The chloroplasts are membranous organelles, just like the mitochondria. They have two layers of envelope around them. The inner membrane closes a space inside it, a compartment filled with stroma, which is the thick fluid. You would find suspended in the stroma are stacks of interconnected membranes. Those interconnected membranes are called thylakoids, and the stacks are known as grana. Within those thylakoid membranes, you would find the chlorophyll molecule, which is the light absorbing molecule in plants. This series of blow-ups takes you into a leaf's interior, then into a plant cell, and finally into a chloroplast. The following chemical equation, simplified to highlight the relationship between photosynthesis and cellular respiration, provides a summary of the reactants and products of photosynthesis. Notice that the reactants of photosynthesis, carbon dioxide and water, are the same as the waste products of cellular respiration and vice versa. That means photosynthesis produces what respiration uses, glucose and oxygen. In other words, photosynthesis recycles the exhaust of cellular respiration and rearranges its atoms to produce food and oxygen. Photosynthesis is a chemical transformation that requires a lot of energy, and sunlight absorbed by chlorophyll provides that energy. Photosynthesis can be divided into two sub-stages. The first stage is the light reactions, in which chlorophyll in the thylakoid membranes absorbs sunlight, and it is then converted to the chemical energy of ATP and NADPH. The second stage is the Calvin cycle, which uses the products of the light reactions to power the production of sugar from carbon dioxide. This is a roadmap for photosynthesis. Carbon fixation is the process of initial incorporation of carbon from carbon dioxide into organic compounds. So you see the relationship between photosynthesis, carbon fixation, and global climate is an ideal example of how interactions between biological components at many different levels affect all life on Earth. Let's start with the light reactions, which involves converting solar energy to chemical energy. What is the nature of sunlight? Sunlight is a form of radiation. Visible light is a part of the electromagnetic spectrum, which is the full range of radiation. Visible light travels through the space as waves. The distance between the crests of two adjacent waves is called a wavelength. Different wavelengths of light appear as different colors. Shorter wavelengths carry more energy. So what colors of light drive photosynthesis? In 1883, a German scientist made an important observation that some bacteria living in water, they tend to cluster in areas with higher oxygen concentrations. Thus, he formulated a hypothesis that oxygen-seeking bacteria will collect near regions of algae performing photosynthesis. He conducted a simple experiment. He placed freshwater algae within a drop of water on a microscopic slide. And now let's see in the next slide what were his observations. As you can see in this figure, Engelmann used a prism to shine the color spectrum of the visible light onto a slide containing algae in water, and he had also added the oxygen-seeking bacteria to the water. His observation was that most bacteria collected around algal cells exposed to blue, white, and red-orange light. Why do leaves appear green? This is because of selective absorption of light by leaves. Now, since energy cannot be destroyed, so the absorbed energy must be converted to other forms. Who's doing the absorption of light? Chloroplasts contain pigments that absorb light of different wavelengths. First type is chlorophyll A, which absorbs mainly blue, white, or red light, and this participates directly in light reactions. Another pigment is chlorophyll B, which conveys the absorbed light to chlorophyll A and does not participate directly in the light reactions. Chloroplasts also contain a group of yellow-orange pigments called carotenoids, which absorb mainly blue-green light. Chlorophyll and other pigments in chloroplasts reflect or transmit green light while absorbing other colors. Chloroplasts are solar-powered sugar factories. They contain photosystems that harvest light energy. Light travels in waves and contains fixed quantities of light energy in packets called photons. The shorter the light wavelength, the greater the energy of the photon. When a pigment molecule in the chloroplast absorbs a photon, its electron becomes excited and gains energy. An excited electron would usually lose its excess energy as heat and fall back to the ground state. In some pigments, the electrons, they get excited after absorbing photons, and as they fall back to their ground state, they release energy not only in the form of heat, but also in the form of fluorescent light. The figure shows excited electrons in pigments, absorption of a photon, as well as fluorescence of a glow stick. In the thylakoid membrane of the chloroplasts, photosystems are arranged and they contain chlorophyll and other molecules. Each photosystem has a cluster of hundreds of pigment molecules that include chlorophylls A, B, and carotenoids. And these cluster of pigment molecules, they function as light gathering antenna. This figure shows a photosystem. The light gathering molecules constituting their antenna focus light energy onto a reaction center. How the light reactions generate ATP and NADPH? Let's try to understand this using the figure in the next slide. This figure shows the light reactions of photosynthesis. If you look at one here, photons excite electrons in the chlorophyll of the first photosystem. Moving to number 2, energized electrons from the first photosystem pass down an electron transport chain to the second photosystem. The chloroplast uses the energy released by this electron fall to make ATP. Moving to number 3, the second photosystem transfers its light excited electrons to NADP, converting it to NADPH. The light reactions occur in the thylakoid membrane. And in the figure in the previous slide, the orange arrows trace a light-driven flow or transfer of electrons from water to NADP, producing NADPH. These electrons also produce ATP. This figure shows how the thylakoid membrane converts light energy to the chemical energy of NADPH and ATP. The light reactions illustrated using a hard hat analogy. The energy of sunlight is used to boost an electron from a low energy state to a high energy state. How the light reactions generate ATP and NADPH? The mechanism of ATP production is very similar to what you've studied in respiration. In both cases, there's an electron transport chain that pumps hydrogen ions across a membrane, and then the enzyme ATP synthase uses energy that is stored by this hydrogen ion gradient to make ATP. The difference is that in case of cellular respiration, it is food or organic macromolecule that provides the high energy electrons, whereas in photosynthesis, the light excited electrons flow down the transport chain. If chloroplasts are solar powered sugar factories, then the Calvin cycle is the actual sugar manufacturing machinery. This process is called a cycle because its starting material is regenerated. The Calvin cycle makes an energy-rich sugar molecule called glyceraldehyde 3-phosphate or G3P using carbon from carbon dioxide, energy from ATP, and high energy electrons from NADPH. The plant cell can then use this glyceraldehyde 3-phosphate as the raw ingredient to make glucose and other organic compounds. The Calvin cycle, let's try to understand the various steps. If you see number 1 here, an enzyme adds each carbon dioxide to a 5-carbon sugar called RUBP. The resulting molecule breaks into 2 3-carbon molecules. Then going to number 2 here, using energy from ATP and NADPH produced by the light reactions, enzymes convert each 3-carbon molecule to the 3-carbon sugar glyceraldehyde 3-phosphate. Moving to number 3, for every 3 molecules of carbon dioxide that entered the cycle, the net output is 1 glyceraldehyde 3-phosphate sugar. The other G3P sugars continue in the cycle. Number 4, using energy from ATP, enzymes rearrange the remaining G3P sugars to regenerate RUBP. Since biofuel manufacturing is highly efficient, biomechanical engineers are trying to solve this problem by taking tips from evolution by natural selection. Now, directed evolution of biofuel production usually uses microscopic algae rather than plants because algae are easier to handle, easier to manipulate and maintain inside the laboratory. In this figure, the researcher is monitoring a reaction chamber in which microscopic algae are using light to produce biofuels. What does a typical directed evolution experiment look like? To begin with, the researcher has to use a large collection of algae, and the algae are exposed to chemicals that produce mutation. Then the algae are screened for their ability to produce the most useful biofuel in the largest amount. So algae that give the best performance, okay, then only those algae are selected and grown, and they're subjected to more mutation and selection. So then after several rounds of mutation and selection, we have the best algae, and the algae may slowly improve their ability to efficiently produce large quantities of biofuels.
Plants provide us with food to eat and oxygen to breathe. They perform this amazing feat by the process of photosynthesis. Let's take a closer look. Photosynthesis requires carbon dioxide, which diffuses into the leaf through small pores and then enters the cells. Inside the cell, carbon dioxide diffuses into the chloroplasts, where photosynthesis takes place. Chloroplasts use energy from light to transform carbon dioxide and water into sugar and oxygen. Zooming into a chloroplast, we see these flattened membranous sacs called thylakoids. Here, light energy is converted to chemical energy in the first phase of photosynthesis, the light reactions. The 2 green structures you see here are photosystems, large complexes of proteins and chlorophyll that capture light energy. An electron transport chain connects the two photosystems. Notice the small mobile electron carriers that shuttle electrons from one large complex to another. Now let's take a closer look at the steps of the light reactions. The photosystem on the left absorbs light energy, exciting electrons that enter the electron transport chain. Electrons are replaced with electrons stripped from water, creating oxygen as a byproduct. The energized electrons flow down the electron transport chain, releasing energy that is used to pump hydrogen ions, the blue balls, into the thylakoid. In the photosystem on the right, light energy excites electrons, and this time the electrons are captured by an electron carrier molecule, NADPH. The high concentration of hydrogen ions inside the thylakoid powers ATP synthase, producing ATPs. The light reactions in the thylakoid have produced two energy products, ATP and NADPH, that will now power the production of sugar in the Calvin cycle. The Calvin cycle takes place outside the thylakoids in the stroma, the thick fluid of the chloroplast. At the beginning of the cycle, carbon dioxide molecules combine with molecules called RUBP. The resulting molecules go through a series of reactions powered by ATP and NADPH from the light reactions. Sugar molecules known as G3Ps are produced. Most of the G3Ps are rearranged back into RUBPs that will begin the Calvin cycle again. But the important product of photosynthesis is the remaining G3P sugar. Some G3Ps are used to build glucose, which can combine into starch or cellulose. Still other G3Ps form sucrose. And some of the sugar is broken down by cellular respiration using oxygen in the plant's own mitochondria, generating ATPs that can power other work of the plant. Excess oxygen diffuses out of the leaf through the pores, while more carbon dioxide enters. With three simple ingredients, carbon dioxide, water, and light, plants produce sugar and oxygen by photosynthesis, powering plant metabolism and ultimately providing your fuel as well.
The thylakoid membrane in chloroplasts contains light-absorbing molecules called pigments. Different pigments absorb light of different wavelengths. One pigment, chlorophyll A, absorbs mainly blue-violet and red light. This molecule participates directly in the light reactions. Another molecule, chlorophyll B, absorbs mainly blue and orange light. We do not see these absorbed colors when we look at a leaf. Instead, we see the green wavelengths that are reflected back from the leaf or transmitted through it.